Hello everyone and welcome to my channel Mrs.Biology with you. In this video series, we are going to discuss about the microscope. So first of all, we'll discuss about the terms related to microscopy like magnification, how image gets magnified, resolution, the resolving power of the microscope and the limit of the resolution. So let's get started. So what is microscopy? Microscopy is a science for investigating small objects using a microscope. So microscopes are what? Microscopes, they are the instrument which are used to observe small objects which otherwise cannot be seen through the naked eyes. So before going deep into how microscope works as an instrument, let's first get started with the need of the microscopy, that is the principle of the microscope. So a human eye, it can only distinguish the things which are 0.1 mm apart, that is about the size of a human egg cell. But the biologists, they want to study these cells which are much smaller than that. For example, prokaryotic cell, which is about 0.1 to 5 micrometers in size, and eukaryotic cell, which are about 1 to 20 micrometers. Now, in order to observe these cells and also to observe the cell biology related to them, our eyes, they cannot see it. Our eyes, they can only distinguish the things which are 0.1 mm apart, and that's millimeters, and here we are dealing with the micrometers unit. So in order to study the cell biology, to study the life processes happening inside the cell like apoptosis, cellular processes, protein synthesis, the biologists, they need an microscopes.
After knowing the principle of microscopy, the two terms which are related to microscopy, that is magnification and resolution, you should know. So here we start with the first word, that is magnification. So the primary function of microscope is what? To magnify the image. Okay, and magnification is what? It describes the enlargement of the object. Okay, whatever the object is in real, the enlargement is what a magnification is. So magnification will give us a magnified image. If you see from this formula, power of magnification is what? It's the power of objective lens multiplied by power of eyepiece. What I mean to say is, over here you have the eyepiece lens, which is generally the constant, that is it remains the 10x. Okay, next is the objective lens. Objective lens, you have the option, you can have 4x, 10x, 40x, 100x. So objective lens has a various option, you can change this, but the eyepiece lens remains constant, which is generally 10x. Since here there are two types of lenses used, that is eyepiece and the objective lens, this kind of microscopes are known as compound microscope. Yeah, and the one which is having only the one magnified lens, they are known as simple microscope. Generally in the lab, compound microscopes are used because there are minimum requirement of two lenses, the eyepiece and the objective lens, right? So let's see how this formula works. Okay, how overall you get the magnification. Say eyepiece magnification is 10x, which I already mentioned that it remains constant, but your objective lens, it has a various magnification. It depends on you on what magnification you guys want to observe your specimen. So if you are observing at 4x, so that becomes 10x magnification of your eyepiece multiply by 4x magnification of your objective lens, which will give you 40x. So your overall specimen which you have kept here, it is 40 times magnified. That's what your magnification is, right? So I hope you are clear with the terminology of magnification. So how actually lens magnifies this image? It is because of the property of the light wave, which is known as refraction. Okay, because of the property of the light wave, that is refraction, this image magnification process works. So what is refraction? It is bending or change in the direction of light when it travels from one medium to another medium. So right here from this diagram, if you see, this is the incident ray which is coming. So it is changing the medium, that is here you have the air and now it is passing through the glass. So whenever your light ray, it is passing from air to glass, so here your incident ray, it bends towards the normal. This right here is your normal. Okay, it bends towards the normal, and also remember its speed decreases, right? When it leaves the incident ray, now when it leaves the glass and it is going again back to the air, that type of ray is known as refracted ray. It bends away from the normal, right? So this concepts maybe you have learned in your physics. So because of property of this, your all magnification process works. That is basically what happens is when this two surfaces of the glasses are not parallel, it will be lens, and this is how the magnification process works right here. Okay, when these surfaces are not parallel, it becomes lens. Okay, and your rays of the light, it goes for the process of refraction when it passes from this glass and it meet at some point where your image gets magnified. So how this lens, it magnifies the image, let's learn from this diagram right here. So here we are going to use some of the rules from the physics, that is from the ray optics. Okay, so let's start from there. So right here is your object. A and B is the specimen which you have kept on the stage. From behind this object, the light source is coming. So these blue ones are the incident ray. So this concept you might have studied in the ray optics, that when the incident ray is parallel to the principal axis, it bends and it passes from the focus, which is F2. But when the incident ray is not parallel, when it passes from the optic center, it just goes straight away and it meets both of these rays, they meet at the A dash. Okay, this ray is not bending because it is passing from the optic center. So right here your image will be formed, but it is still inverted, real, and it is smaller than the object. Our aim is what? We want the magnified image. So we have kept for now the object beyond the F, that is beyond the focus. Okay, so now what we will do is now we will keep the specimen at 2F, that is we are trying to make this specimen closer to the lens. Okay, so again your image, what happens is when you are kept at 2F1, the same rules applies and your image is formed at 2F2. Still it is real inverted and it is same size. Now the size is same. In the first diagram the size was smaller, in the second one right here we have the same size as that of the object. Now let's see what happens when we are keeping it between 2F1 and F1. Your image is formed beyond the 2F2. But here you have the larger image. So I hope you have noticed over here that as we have decreased the distance between the specimen, that is your A and B, the object, and the lens, your image gets bigger. but still it is inverted. This one right here, you will get your image magnified, real, magnified, virtual and erect, but behind the lens. Okay, when you are keeping your object between optical center and F, that is between F and O, when you have kept your specimen, you will get an image behind the lens, not here. So when you extrapolate this light, here you will get your image, which is virtual, erect and larger than the object, which is our main role, where we want the magnified image. So let me show you this whole scenario with the ray diagram. Okay, now compare this whole thing. Here is your object. Your objective lens will make an initial image right here. Okay, so this initial image will work as an object for eye lens. Later on, your eye lens will make the image, final image behind your object right here, which will be magnified. So all these rules of ray optics which we have studied is applied over here. So first one, what is happening is objective lens, it is making an initial image for the eyepiece lens right here. Then your eyepiece lens allows the final image, which is virtual, erect and behind the object, final one, magnified one. Okay, so see, like this. When you have kept your object here, first of all your real image will be formed through the objective lens. This real image will work as an object for eyepiece lens, and then later on eyepiece lens will form a final image which is virtual behind the object. That's what you see, the magnified image. So this is how the image magnification works. It has some of the ray optics and physics concepts. Your magnification is like infinite, but at some point you cannot gather the information from a magnified image, and there comes in picture the resolution, wherein you can have the fine detailings of the picture which you are observing. That is the fine detailing of the specimen which you have kept on the stage to observe under the microscope. So let's discuss what is resolution. It's an ability of a lens to separate or distinguish between two small objects that are very close to each other. That is, I'll explain you through this diagram. That is, this is the objective lens. Okay, this objective lens, the ability of this objective lens to resolve this object, okay, which is placed under the microscope over here. Okay, so you see they are very close to each other, but how finely this objective lens can distinguish between these objects, that's what a resolution is. So this is the best resolution obtained. Okay, and this is the poor resolution, wherein you can only see one image. So when your images are not resolved, you will observe it as a one whole object, one whole point. Okay, when they are perfectly resolved, you can see that they are actually two blocks or objects which are placed. Yeah, so that's what the resolution is. So now whenever you are choosing a microscope for the study of cellular activity, resolution is taken into consideration, because resolution will lead us to the fine detailings. Okay, fine detailings can be known. So your resolution is important whenever you are choosing a microscope. But there are some factors which affects this resolution, that is the resolving power of the lenses, which are these four factors. So you can take a moment over here and you can just make a note of these 4 points, which is wavelength of the light, refractive index, angular aperture and diffraction limit of the light, which we will discuss each of it in detail. Okay. So first one is wavelength. So your wavelength is inversely proportional to the resolving power. That is, if your wavelength is shorter, higher is the resolution. If higher is the wavelength, shorter is the resolution. Why? Because limit of resolution is equal to the wavelength, and the limit of resolution is minimum distance between two objects. So if your wavelength is high, your resolution will be low. If your wavelength is low or shorter, you will get a high resolution. So I hope you are getting it what I'm trying to tell. Your shorter the wavelength, higher is the resolution, and longer the wavelength, less is the resolution. This point you will also get to know when we will discuss about the diffraction, how diffraction also affects the resolution. So your wavelength is inversely proportional to the resolution. Shorter the wavelength, higher is the resolution. Okay, and that's the reason in electromagnetic spectrum we are only using the visible light, so that we can get the better resolution. Yeah, so this is how the wavelength affects the resolution. Next is your refractive index, which you can represent it as a Ri. So more is your refractive index, the more is the resolution. For simplification, you can take refractive index as a optical density. So this is the main reason we are using the oil. Immersion oil are used in observing some of the specimens. Why? To increase the optical density. When you place on your stage, you have kept a specimen. On those specimen, you have dropped a oil. One droplet of oil is placed. This droplet of oil will increase the optical density of your specimen. And if you see, you have increased the density, the rays, they are bended, and they all are gathered and they are going right upwards. If you are not placing an oil, you see your waves, they will just diffract more. Okay, here is your specimen, say for example, and here is your lens. So you see, when the rays are coming, they will diffract more. Only this many rays are gathered, and from this many rays you will get an image. This is without oil. Now when you have kept an oil, what happens is light with, okay, here. Now you have kept oil, what happens is all the rays will diffract less and you will get everything perfectly on the objective lens. So you see here it's with oil, the path is with oil, where diffraction was less. So lesser the diffraction of the light waves which were coming from the source, all the rays has been absorbed by this lens. Okay, and this rays will help in what? Help in the formation of the image. So refractive index, you can simply take it as an optical density. So more the optical density, better is the resolution, and that's the reason we are using an oil. Many of the students have done it, an experiment taking a drop of the oil on the specimen to increase the resolution. This is the reason. When you keep an oil, the optical density of the specimen will increase. Your rays of the light which are coming, they will diffract less, diffraction will be less, so that more of the rays will reach on to the object, and that's where you get your image better. So third one is the angular aperture. Okay, so what is an angular aperture? It's an angle which is formed between a specimen and the objective lens. So you see this one, it's an angular aperture. It's what? It's an angle which is formed between the specimen and the objective lens. So higher is the angular aperture, the better is the resolution. Okay. So how you can increase the value of this angular aperture? So there are two ways to increase the value of angular aperture. That is the angle between what? Between the specimen and the objective lens. So first one is when you decrease the distance of specimen and the objective lens. So when you decrease the distance, your angle become higher. Okay, and second one is you can use the convex lens thick. So thicker the convex lens, the higher the angular aperture, and the less the distance between the specimen and the objective lens. Distance is less, and therefore you will get the higher value of angular aperture. So higher the value of angular aperture, better is the resolution. Last one is diffraction. How diffraction limits the resolution. That is, if you see from the figure, you will also get to know the concept of the wavelength over here. You see blue light which has a shorter wavelength. So shorter the wavelength, the less it gets diffracted and the more resolution you will get. You see over here, here is your specimen. This smiley is your specimen placed on this slide, and here are your light rays which are coming. So when you see, when they hit it on the specimen, they are diffracted less. All of those are gathered back into the objective lens, which will get you to give a better resolution. So here you can also relate the concepts of the wavelength. The blue light has the shorter wavelength. The shorter the wavelength, the more is the resolution. Also, when you are using a shorter wavelength light, your diffraction will be less. And the less the diffraction, the more you will get the resolution. When you compare this with the same thing with the red light, they have the longer wavelength, theta is high, so you see the rays are diffracted more. They are not all gathered into the objective lens. So when these light rays, they are not getting into the objective lens, how the image will be formed? Okay, and if image is not formed when they are diffracted more, resolution will ultimately be less. Okay, so here you see same thing. Blue light, it gives a best resolution, the distance is best, but when you are using same thing with the red light, you see the distance you are seeing, they are quite closer to each other. Here also see, it's a poor resolution with the red light and best resolution with the blue light. So diffraction also plays a major role. So I hope we are clear with the factors affecting the resolution and what is the resolution. This will help you to understand the types of the microscopes which we will discuss in the next video. So thank you very much guys. I hope it has been a helpful video to you, and if you have liked this video, do subscribe my channel and click the like button, and also stay tuned for the next video, which is on the light microscopes, electron microscopes, working of them, advantages and disadvantages. But mainly we'll discuss about the which microscopes has the better resolution and which will be more helpful in getting you the fine details. So stay tuned.